hydrogen bonds between what groups? So we understand that tertiary and quaternary structure have, non-covalent interactions between the R groups, including disulfide bonds.
Sorry.
But secondary structure.
You want me to answer?
Yeah.
Okay.
Secondary structure is also disrupted because it has hydrogen bonds between the polypeptide backbone. So that's important to remember that tertiary and quaternary structure. Tertiary structure is a single polypeptide subunit. Does she talk about subunit and domains? Yeah, she talks about subunit, the difference between subunits and domains. So tertiary structure is a polypeptide subunit, one polypeptide, and it represents the R group interactions between the side chains. Quaternary structure has multiple subunits, so multiple polypeptides, and it's those interactions between the polypeptides is what quaternary structure represents. So by increasing the temperature, we're going to disrupt those non-covalent bonds, so those hydrogen bonds in the polypeptide backbone and secondary structure, the non-covalent interactions in tertiary and quaternary structure, and like you said, in primary structure you have the polypeptide, the amide bond. So those are covalent bonds, and since it's a moderate increase in temperature, those bonds are likely not to be broken. Another thing that we don't have on this slide is that tertiary and quaternary structure do have a type of covalent bond. There's a special situation where if the R group has a cysteine residue, it has a covalent bond. There's covalent bonds between those cysteine residues, and that's called a disulfide bond. So those disulfide bonds would also likely not be affected by a moderately high increase in temperature because they are covalent bonds. So those are really important for maintaining quaternary and tertiary structures because they form disulfide bonds. Does that make sense?
Yeah.
For the disulfide bonds, are those in both tertiary and quaternary?
Yes. Yeah.
Anything that's R group is tertiary and quaternary. If you guys remember, one of the questions on the quiz asked you about two of the R groups in that chain that was provided and what levels of structure are involved. So some people were saying secondary, and the reason it's not secondary is you have to remember it's only backbone interactions with secondary from topic 1. So just keep that in mind.
Any other questions? We have a question for you.
Yep. I guess this is another thing on a worksheet. This is more of an application question. So think about it. You might have to apply concepts from last topic, but try and answer it to the best of your ability. And then if you participate, I'll give you candy.
Good incentive. Good incentive. Just have them participate in that too.
Exactly. Awesome.
Thank you. Do you buy it?
Yeah, it was like $4 at CVS.
Shit. I could have just got my friend to get it from ILC because there's like whole ass like
Bags?
Yeah, no, they'll just like you can just grab a bag and just put candy in there. It's for like SIs, but like, you can just put like
Same thing.
Yeah.
Oh, I thought you meant like there was like a bowl of candy.
No,
I was
no,
like,
no.
nah,
They
it's
have
fine.
like a whole shelf, and there's like lollipops and like Starburst
Wait,
and like
bro.
Jolly Ranchers. And just,
I'm gonna do that next time.
yeah. It's where ours is. I don't know
Yeah,
what
floor 12,
yeah.
12, yeah.
I think it's better if I don't switch the answer and just let them like discuss it.
Yeah.
Because I'm figuring it out.
Yeah, no, you got it, you know, first session. What the heck? A dice with the actual numbers on it.
Some freaky shit, bro.
Yeah.
We'll keep going quick. We got time. Yeah, we got time.
It's good. We're gonna just let him spend time on this. All right, if you want to take the time to talk to your neighbor about the question, if you aren't already, go ahead, share your ideas, and then we'll come together as a big group.
You should look at like what they're being taught too. I didn't take this class. You should look at like what they're being taught, like lectures and stuff.
Yeah, you're fine. I mean, you have the knowledge. It's just like
Yeah, it's just like
which way to apply it.
Yeah.
I got you. Not really that deep.
I think it's okay. I mean, if you share it, it's sure.
Yeah, whatever.
It looks like he's doing other
I don't think any of this.
All right. Who's got some ideas? Who wants to go first?
No ideas.
I'll start calling tables. Yeah.
For A, kind of just said like the maturation of the proteins can happen because if we talk about environmental factors
Huh?
that can cause like the non-covalent bonds to vibrate more stable
Mhm.
or so it could be like thermal energy, pH,
Okay, I'll ask you a follow-up question, putting you on the spot. So, what do you think could happen in the cell that causes the pH to go up, or that causes it to be, I don't know, too hot? Do you have any ideas?
You're like sick if you're like a fever maybe.
Yeah, that could be one thing. Or like, too many ions go into the cell, and if too many hydrogen ions go in, what happens to the pH? It goes down. Yeah, there you go. So those are things that can happen to cells. Like random ions can go in, and if it happens too much, the pH goes down and disrupts cell processes. You want to catch it?
Good throw. That was a dot.
All right. Anybody else for A? Any other ideas? We good? Yeah? Yeah?
Oh, no, I have a question.
Oh, yeah, go ahead.
Oh, yeah. I guess the aqueous environment cell obviously also affects if the proteins unfold, then hydrophobic elements which will come together, and that might not be in the way that they are supposed to form.
Mhm.
And then hydrophilic elements are gonna want to come outside of that aggregation, and that's obviously gonna lead up to like a toxic buildup of those proteins.
It's good. Yeah.
Yeah.
Was there a
Already
question?
hinting at question 2 as well. The second part of this question. Does anyone else have? Or you had a question?
Yeah. When we have questions like this, like on exams or quizzes, when we talk about environment, like I'm thinking of like the environment in this like an aqueous solution, or like maybe like one that's more hydrophobic. Are we talking about like temperature and things like that, like temperature, vibration, or are we talking about like aqueous and like hydrophobic, hydrophilic?
It will always be talking about the aqueous environment of the cell, and so like with temperature, like random things can cause that aqueous environment to heat up, and because it's getting hotter, it'll vibrate more. Like stuff inside that aqueous environment will start to vibrate more, and then that'll lead to these other effects.
So the difference between the environmental and cellular conditions, like the environment would be if there's increasing pH, then it would create as well. Cellular condition would be like how the hydrophobic groups like are they oriented, or what would say?
Well, it's talking about like the environment of the cell. So So we're talking specifically about, because cell protein folding happens in the cell.
Yeah.
So when we're talking about temperature increase, that's temperature increase in regards to the cell.
Okay, so that would be cellular conditions.
Yeah. Or it's all considered like environmental.
Okay, so you can kind of group those two together.
Yeah.
environmental and stuff. Okay.
Yeah. Like if your body temperature increases, obviously the cells
Yeah.
are the temperature of the cells are gonna increase.
Got it.
Yeah. Anybody have anything else for A? Or should we move on to B? Okay.
Alright, any takers for B?
So this was hinting a little bit at what I was talking about before, about aggregation.
I know like this has to do with like chaperones, because well chaperones can help stop it, but when the protein unfolds, basically then the hydrophobic core is exposed into the aqueous solution, and it's gonna do anything possible to not be surrounded by water.
Mm.
So other proteins that are unfolding, they'll all like kind of come together and get stuck to each other.
Mhm.
Just like you said, that the protein can't refold because they're like our groups making interactions.
Yep.
Yep.
Sol?
Yeah, I think that's a good explanation. I would just say, what do you think happens with individual amino acids? You have the perfect idea for what happens at a protein level because it unfolds. So, does anybody have an idea of what happens to each, maybe not each specific amino acid, but what changes between amino acids when things unfold? Yeah.
Either combined other like salts in the environment or the vibration to them causes them the bonds to break between two R groups,
Mhm.
which causes it to deviate and form immiscible.
Which bonds would break?
The non-covalent bonds to two R groups.
Yep.
Anything else you had, Han?
Is there like any change in shape and change of function during this time?
Yeah.
Yeah. So if it unfolds from a clump to like a strand or closer to a strand, it's going to change its structure. And if you took 151 with Laura, then the biggest thing she harped on was structure equals function. So just remember that if anything involves unfolding or refolding, it involves structure, which will involve function.
I think a big part of this too with aggregation as well, if like I know the first sentence it doesn't really have anything to do with like this question right now. But this is a good example of like an application question that you guys can get on an exam where she'll ask you to explain how does aggregation at the cellular level affect a disease like Alzheimer's, or there's Creutzfeldt-Jakob as well. And being able to explain how the proteins, how the hydrophobic interactions occur with each other, how that process of aggregation causes cell death, causes tissue death, results in neurodegenerative disorders. And beta amyloid proteins into those like plaques. Those plaques are what we see as protein aggregates. So that's a direct result of those proteins coming together and aggregating away from the aqueous environment. Does that make sense? Okay. So this is just the answer. We sent this out to you guys in the slides as well, but you guys pretty much hit it on the dot. pH, heat, oxidative stress is sort of like cellular conditions, just random chemical processes that can happen in the cell and disrupt protein folding, but I don't think you guys need to know that as much. Hydrophobic clustering away from the aqueous solution, the hydrophobic effect. Does anyone know why the hydrophobic effect is favorable at a chemical level? This might be beyond the scope, but this is helping explain it.
Laura could ask that. You never know.
Yeah.
So why do you think, like, if something unfolds, why do you think it would try to couple with something else that's unfolded?
Yeah.
And then, in turn, this is the reason why it's called the hydrophobic effect.
Yep.
Like when the protein unfolds, the hydrophobic R groups are exposed to the aqueous solution, and so they just start like hydrogen bonding with anything nearby that prevents it from then having a contact.
I would just not say hydrogen bonding because the hydrophobic R groups probably are not involved in hydrogen bonding, but yeah, they would want to, hydrophobic R groups want to stay in the middle or like the inside of the cell, and so if they're exposed, they're now on the outside, so they'll couple with something else that's hydrophobic, and now together, all that hydrophobicness is now on the inside.
Yeah. And so another way to think about it is that being those hydrophobic groups clustering together instead of being exposed to that aqueous solution, that's thermodynamically favorable for the cell. So the cell wants to, keep their, did they talk about delta G? The cell wants to keep their free energy or delta G negative. So they want to keep this process thermodynamically favorable. And so they want to cluster those hydrophobic R groups together and the hydrophilic groups on the outside. That's why in a globular protein, you have the hydrophobic groups in the center of the protein and the hydrophilic residues on the outside interacting with water.
Because it decreases the free energy.
Yeah, it makes the process more thermodynamically favorable. Okay. Any questions on this?
All right, moving on to heat shock response and unfolded protein response. These are like, as the title say, responses to these different environmental stressors. So heat shock response happens in the cytosol and the nucleus. There is a mechanism that you don't really need to know the specifics of, but it senses that proteins are being misfolded. And so those get sent to the proteasome, which you can think of as like the garbage disposal of the cell for proteins. When those proteins are being unfolded or are in the process of being unfolded, chaperone proteins might be able to help out and refold them. It's not like a 100% like success, but they are involved in like maintaining some sort of stability. And during this whole response, the chaperone proteins are upregulated, which means there are more of them produced, so that they can try and help with the mass unfolding that's going on, which is what Anike talked about earlier. So the second thing is the unfolded protein response, which happens in the endoplasmic reticulum. It senses the unfold. Again, broad terms, you don't need to know the super in-depth mechanisms, but ER chaperones are upregulated, so this is very specific to the ER, whereas the heat shock response is mainly in the cytosol and nucleus. But on the other hand, with the ER, the protein has to leave the ER after the chaperone's done, and it retrotranslocates to the proteasome afterwards if it's not properly folded. So, main idea: if it gets folded, it's because the chaperone helped out. If it didn't get folded, it's sent to be disposed of in the proteasome.
Solid.
And so then a quick overview of just protein functions and mechanisms, and oh, you hear about?
I just have a question about outside.
Yeah.
What does upregulate from deregulate?
Oh yeah, sorry. So upregulate means after something. This is very general. So after something happens in the cell, there could be a need for certain proteins more now. So like let's say a protein exists. Let's say there's 10 of them in a cell. But after a certain condition has happened or some sort of event has happened in the cell, now you need 20 of them. The process of recognizing that and creating new proteins to reach the desired amount is called upregulation. Versus an event could happen where you had 10 of these proteins and now you only need 2 of them. And so removing the excess proteins or not making more of them is dysregulation. Or sorry, downregulation, not dysregulation. Yeah. And then so with function and mechanisms, you can kind of understand these in the whole broad view of unfolding as well. And then as I go through these, think about if unfolding happened, what would be disrupted with each of these. So with ligand substrate complexes, the basic thing here is a substrate is just another thing. It's usually a protein that binds to, or a ligand is also a substrate. So these things are something that binds to other proteins, which achieves a certain function. So if you ever see Laura use ligand or substrate in a problem set, just think of it as a protein that's going to bind to something else. Protein ligand affinity is describing the relationship between that ligand and the protein that it has to bind to. High affinity means it is more tightly bound, more strongly bound, and a low affinity means it's more weakly bound. And the way these bindings and like connections happen is stuff moves randomly in the cell. There's no rhyme or reason to how it moves, it just moves. And eventually these substrates and proteins will come around and then bump into each other, and if they have a high affinity, they'll stick. And yeah, they'll form non-covalent bonds, though. That's the most important thing. Covalent bonds are like actual like connections, but non-covalent bonds, as you guys know, are more like attractions. And so there are things that can disrupt protein ligand affinity, which I'll go over, but the reason why they can disrupt those bonds is because those bonds are non-covalent. You guys remember why covalent bonds are the strongest in the cell, right? Thumbs up. And then lastly, the relationship that the thing that you can use to describe the relationship of these bonds is the dissociation constant, Kd. It means how likely that ligand is to dissociate or separate from the protein it's bound to. Yeah. So there's a formula for Kd. I'm not sure that you need to remember, but we threw it on there just in case. Usually if you have like a formula on a test or something, Laura will give it to you, but on the off chance she doesn't, just try and remember that. Yeah.
What if she asks you to explain the relationship between that equation?
I don't think she's going to explain that.
I mean, it's also good to conceptualize. Like, if you know the equation, you can know like an increase in what happens if there's an increase in the concentration of the complex. What happens to the Kd? What happens if there's an increase in the concentration of the protein or the concentration of the ligand? What happens to the Kd? Just by remembering, you know, the equation and the relationship between the protein, the ligand, and then when the protein and the ligand are bound. So when the ligand occupies the binding site of an enzyme. Just understanding how that affects the dissociation constant is important and can be really helpful on exams and stuff.
It's a good point, because what you can kind of conceptualize is if there is more complexes, like the C over there, if there's more complexes, the Kd will go down. We'll get into what that means because there's a question about it, so I won't explain it just yet. But just think about like if there's more ligands in the whole, just like plain ligands in the whole cytosol, the Kd goes up. But if there's more complexes, the Kd goes down. So just think about that as we go to the next one.
Anyone have any questions right now? Should we go into? All right.
Yeah. So again, to reiterate, this affinity needs a right balance of everything. So again, the Goldilocks hypothesis is brought up. I think I mentioned earlier, when you have a bond between a substrate and a protein, if it's too hot, or if the bonds holding the protein together are too tight, then that substrate can't bind, it can't fit, right? So too many bonds or too strong bonds are, again, not necessarily better. It needs to be just right. So just keep that in mind when you're answering problem set questions or exam questions. Just remember that. Okay, question, I think it's on your worksheet, double check. But what determines protein ligand affinity? So you guys can answer that. Think about like what kind of things on a protein, what kind of maybe bonds affect affinity, and then if that protein's changed such that the Kd, the dissociation constant, is lowered, what does that mean about its affinity? Take like a minute, and then after you thought about yourself, talk to your neighbor. Who's got answers? Yeah.
I wrote down that the chemistry of the R groups affects the affinity between them, also the shape and the size of the ligand and the binding site. So if one is too big or too small, it can fit. And also said like the needs of the affinity. So if the affinity needs to be higher, then it will be higher. If it doesn't need to, it will be lower. And then the second part, it has a lower KD, the affinity will be higher since KD measures how likely it is that they will dissociate.
Does that make sense, everybody? Good explanation. Yeah.
That's okay.
Yeah, so pretty much hit that spot on. Affinity is determined by the number of bonds and what kind of bonds exist, let's say in that active site. So if there's a, I don't know, I guess actually that's the next question, so I won't just say just yet. But yeah, and then KD, think about it in the opposite. So higher KD, lower affinity, lower KD, higher affinity.
I think it's also important to remember that, like, when we're talking about proteins, ligands, as Shoria mentioned before, everything in the cell is essentially spontaneous. So these proteins and their ligands, a good example is like an enzyme and its substrate, what reaction it's going to catalyze, and I'll get into that later. But having those enzymes, their active site, where they're going to bind to their substrate and where they're going to perform like their catalysis reaction, the ligands and the enzymes are just floating around the cell. And those will bind to each other through random non-covalent interactions. So interactions that we talked about, hydrogen bonds, van der Waals interactions, pH, charge. So having that perfect match will allow for that catalysis to occur. And I'll talk about this later in the slides too. But I think that's a good point that Joria brought up that I wanted to reemphasize. Okay. We have another application question for you guys, so take a moment to read, write it out. It's good practice for the exam and the quizzes. And then talk to each other, and we'll regroup.
Yeah, so take a couple minutes, work on it yourself, and then after a couple minutes, talk to your neighbors, discuss, come to a understanding.
Have a good one.
Have a good one.
They usually ask like problem set questions too afterwards. Do they usually come up and ask like problem set questions?
At least in 151.
I think so. Because those are like, not like intro, right?
When are your office hours? Oh wait, I was just there the other day.
Thursday to 4. Do we put that on the slides?
It's on the campus.
So she moved all my shit for me, so I don't know where it is. Dude, these were my mom's headphones. Because she asked me, she found them last week. Thank God, because I just been brand new for Christmas. I was like, Mom, it's important. And she found them, but she didn't even sit there until she found them. Anyway, let me.
it says like this position is involved in binding the extracellular ligand. Is it increasing or decreasing binding? It just says it's involved in binding.
That's
Think about it as like the amino acid at that position is directly in the active site and is directly like gonna bind to something on the ligand. Yeah.
Might have been helpful to take like a map.
Oh yeah, picture.
I mean, she gives you guys that on exams, right?
Oh yeah.
Yeah. So for 423, we need to memorize them. For 423, we need to memorize them.
What?
The amino acids.
Oh yeah.
I mean, for MCAT too.
Yeah.
It's good practice.
You download amino acid quiz.
I'm on my phone.
Oh yeah.
I've been doing it every morning.
Every morning.
So clutch.
Rehearse it in the shower. Like. Oh, so you're officially started studying, or not really?
Well, for 423, because it basically just covers the biochem section of it. So, it's honestly, I might start studying like with it and then after it.
After it's done, yeah.
I think studying with it to know like what I should look for in MCAT,
Oh, yeah.
and then like understand like, because I learned it really in depth, so
Yeah, that's true.
it's helpful. That resource spreadsheet that you gave me too is like amazing.
Isn't it has everything?
It's everything you need.
And so like obviously you have to buy some stuff, like you have to buy URL, but
Yeah.
but all the free stuff on there is like
So clutch.
Yeah.
It's so clutch.
Because there's like so many free practice tests just
Yeah.
like scattered.
Are they accurate? Do you think they were accurate towards like the actual one?
I'll show you which ones are best.
Mine comes back.
I would lean towards the ones that are harder.
Gotcha.
The easier ones will.
You want to get your expectations too high.
Oh yeah. Always tamper the expectations.
Gotcha.
You got a 520? Nah, you didn't. On to get on the real one.
It's like automatic 5-point deduction
Yeah.
from whatever you get.
It's like people, it works the other way too, because people have gotten like 508s on practice tests and then get 517 on the real one.
It's crazy.
Or they get like 523s on the practice test and get like a 515 on the real one.
Yeah.
So like, alright, who's got some ideas? You can answer one part, all parts, whatever you want. Yeah.
You'd be going from negative to a positive charged one for amino acids, so it's going to change the interactions that it's going to make with other amino acids and other R groups, which if you needed to do a specific thing and then it needs to make those specific bonds, it's not going to do that anymore, change it up. So there's going to be a low affinity and a higher heat.
So, let me ask you a follow-up question, right? Something important to we should have probably displayed this, and she's gonna give it to you on exams. Is like the amino acid chart, so it'll show you the side chains of the amino acids. So when you're changing the glutamic acid to a lysine, what part of the side chain is changing here? Yeah. The R group, but like exactly. So do you have it pulled up? It's important for this question specifically. Yeah. So we're changing. The glutamic acid has a carboxylic acid group, COO minus COOH, which has a negative charge, and you're changing that to the lysine's positive charge, which is an NH3 plus. So if you change a negative charge to a positive charge, what's happening now to the binding between the extracellular ligand and the binding site at that specific amino acid?
Yeah.
Yeah.
And so because it's pushing it away, or you can also think about it as if it normally is glutamic acid, what's probably the charge of that complementary position on the substrate or the ligand? Positive, right? And so now that the glutamic acid changed to something positive, it's going to repel the other positive one that's on the ligand.
So for those of you guys who haven't taken Laura before, this is sort of the level of explanation that you need to think about when you're writing out your explanations to these questions, because she has all free response questions.
Mm-hmm.
So it's important, and this is also something she can ask you as a follow-up question, as like a part B on an exam or a quiz. So it's important to look for those details as well. Does anyone want to talk about the KD of RTK, how that changes?
Or you can just talk about there's a change in the charge, so it's not going to form as many bonds as it was before, so it's going to increase the KD as it's more effective.
Awesome.
And then lastly, like with the little blurb I gave you at the beginning, what does this whole new change to lysine do to the whole pathway? This is like a broad thing. You don't need to know the specifics. Yeah.
So the like binding of the ligand, it regulates cell division. So if it's unable to do that, then the cell will divide, and that will decrease like or overall in the body, like cell division will decrease, and then that'll probably have an effect on cell life as well as cell health.
Exactly. Yeah. Exactly. And so that's another point. You answered that great. Another point that you should remember is most, if not all, of these questions will have like a bigger picture concept that you might not need to know perfectly, but you need to know that like, okay, here's a change that happened. Here's what happens later on in its processes, and here's what it does to the bigger picture concept. So you need to write all those things in your answers on exams or quizzes. A lot of people answered. All right, you can go next.
Yeah. So this is more or less of what you guys talked about. Yeah. A good point to mention also for the last blurb of this question is that if you have, if the cell is unable to divide, then there will likely be. Have you learned about apoptosis?
Yeah.
Yeah. So if the cell's unable to divide and the cell can't divide, there's likely to be signals for automated cell death, which the cell will basically self-destruct, and that's called apoptosis. Not mentioned here, not mentioned in the answer, but just something important for you guys to know. And you'll learn about that in later units as well in cell division. Okay, next question. All right, so now I touched on this a little bit earlier, but looking at enzyme function and mechanism. So enzymes are proteins, as we know, as I talked about. They have an active site, a binding site, and these proteins can change their conformation or change the way that they're shaped around the ligand, around what they bind to make or break covalent bonds. So normally when you have a reaction in the cell, what's an example? You're forming a peptide bond, right? When you're forming a polypeptide chain, you're forming that peptide bond. The amount of time or energy that takes for that to happen spontaneously will take, forever. For those molecules, for those atoms to be in the perfect conformation or the perfect position to form that peptide bond spontaneously without the help of anything else, it's never going to happen. So that's why we have enzymes. Enzymes work to reduce this time, reduce this activation energy, and allow for that to occur at a faster rate, easier rate. And these enzymes basically allow for all of these cellular processes to occur. This is really important. That's why we need proteins. So, as I mentioned, they'll stabilize that transition state of the reaction. So the amount of time that it takes for that products to transition to, or for reactants to transition to products. And if there is a spontaneous or exergonic reaction, then the product energy will be lower than the reactant energy, making that process thermodynamically favorable, versus a non-spontaneous reaction, which means that the product has a higher energy than the reactant. As I mentioned before, enzymes will bind to the ligand, and they'll remain unchanged at the end of the reaction. So they'll change their conformation around the substrate, and they'll allow for that catalysis, for that reaction to occur, and then they'll change back to their original conformation and wait for another substrate to spontaneously come into the enzyme. Yeah, and they won't have affinity for the product. This process, they'll dissociate. In ribozymes, which is in the RNA/protein part, this is an example of how catalysis can occur in protein formation, for example. So in translation, which you guys will hopefully have learned about and will learn about more.
Can someone shout out why it would be bad for the enzyme to have affinity for the final product?
You want to dissociate quickly.
Was that?
You want to dissociate quickly.
Yeah, I mean, like if it had affinity for the final product, then it would just never dissociate from the enzyme and never go out and do what it needs to do. Another way to regulate protein function, and I know we're kind of going short on time, so I'll go fast, but allostery, if you know the word, you know it means binding kind of elsewhere on the cell or on a protein. But one thing I mentioned earlier was protein function is always related to its structure. And so if you bind somewhere else allosterically on that protein, it's going to change its structure and then change its function. And so that's how it can be regulated. It can be upregulated or downregulated. Like it can increase its function or decrease its function. Basically, like when we say conformational states, it's just another different structure of that protein as a result of something binding to it. So with allosteric binding, they adopt new conformational states. And then one thing that you'll go more in depth on in like topic 5 or something is dimerization. It's basically just when two proteins come together, and usually that's as a result of an allosteric thing that binds to it. So the other question that that I asked earlier was the RTK pathway. That has a lot of dimerization happening to it. So keep that in mind. It's basically just two things being connected as a result of allostery.
Yep.
Another thing is post-translational modifications. So the biggest thing to remember with these are covalent modifications. And all this time we've been talking about non-covalent interactions and non-covalent binding and all that stuff with affinity. But these are covalent modifications that require a lot of energy to happen. And because they require a lot of energy, they are facilitated by enzymes. So it's another thing to remember. Like anything that requires energy or that needs to be made with a covalent bond probably needs enzymes.
Yep.
So you can reverse these two. Those same enzymes or maybe some different enzymes can take off those modifications. Usually, like the biggest thing you'll see is phosphorylation. A phosphate group can be added, but can also be taken off. So phosphorylation, or the reverse, is called dephosphorylation.
And a good point to remember about this is that just, talking generally, when you have a protein, when the protein folds, it's not all the proteins are gonna perform the exact function that you want them to perform, right? It's not gonna always do the same thing. There's a lot of modifications, post-translational modifications, transcriptional modifications at the DNA level, which can subtly tweak and fine-tune these proteins and their functions. So a good example is when Shoria talked about heat shock response, and how the proteins that are misfolded or not properly folded are sent to the proteasome for degradation. How do they get sent there for degradation? Post-translational modifications. And that's through ubiquitination, which I won't go too into depth about, but just know why these are important as well.
Is phosphorylation because it needs a kinase to bind? Is when a phosphate binds to a protein, is it a covalent bond?
Yes. And that covalent bond, it'll change the overall charge at that amino acid, which will change its conformation, which will change its structure, which will change its function, which is something Laura really likes you guys to explain and over-explain about.
The last the last of the three ways it gets regulated, or proteins get regulated, are through nucleotide binding and hydrolysis. So you guys know what ATP is, but there are other molecules like GTP. The A in ATP is adenosine, like the nucleotide base. G is guanine. So those are two different structures, and ATP can become ADP. You guys have probably heard that before, but similarly, GTP can become GDP. So anytime those are mentioned, like you can kind of think of those as a similar thing. So enzymes can hydrolyze the third phosphate in ATP to become ADP, or in GTP to become GDP, which will control function of those proteins, basically. So ATP aces, when we say ace, it's like another word for enzyme. So an ATPase or a GTPase will affect the ATP or the GTP. And then these ATPs and GTPs will go on and bind other things. So if you have a big enzyme that's bound to GTP, it could be called in the active phase. But if it's bound to GDP, it could become inactive. So this is kind of like another level of the post-translational modifications. Think of ADP versus ATP as two different sides of like a ball. And if it's bound to one side, it can activate a protein. If it's bound to the other side, it could inactivate the protein. Yeah.
And these ATP and GTP, they're carriers of energy. So enzymes will use these, the release of the energy when you break a bond, to catalyze a reaction. Any questions?
Yeah.
When it releases the phosphate molecule, does it also release energy? Like, is that the purpose of the GTP?
Yes.
Usually anything that involves breaking a bond, like in this case, breaking the GTP to a GDP, it's going to release energy.
That release of energy is coupled with the enzyme is using that release of energy to catalyze the reaction. And then once the energy is all used up, the new substrate is left over. Any other questions?
All right, last one. This is a little hint on to problem set 11, or sorry, problem set question 11. So we talked about how post-translational modifications can regulate proteins. So can somebody explain how specifically it causes a conformational shift using all the big words that Laura likes to hear? Take like a minute, because I know we're kind of running short on time, but take a minute and then I'll call on somebody. And just a quick reminder for the question 11 on the problem set, you're going to need to talk about all three regulation mechanisms, not just post-translational modifications. Does anybody have any ideas, any guesses? Someone who hasn't spoken up yet?
We have candy.
Yeah.
It's a good incentive. Really don't want a cold call.
Something about structure? Does anybody want to say anything about structure? Yeah.
Okay, I don't know if this is right, but I said that like something like if you add like a phosphate group, and when you do that, you're changing like the structure of the protein, because like you're like adding the phosphate group, and when you change the structure of the protein, like you change the shape, and then because it's different shape, like the protein would have like a different function. I don't know if that's like.
Yeah.
Yeah, no, that's perfect. If you think about it, like phosphate groups are really heavily negatively charged. Like if you just Google a picture of a phosphate group, it has negative charges on it. And so when that gets added to somewhere on a protein, it's gonna affect the surrounding area where it got added. Because it's so negative, like let's say it's near an amino acid that's positive, those will start to kind of push closer together. So let's say you have a residue of lysine, which is a positive one, and then the phosphate group gets added to a nearby amino acid. The shift might happen because those two amino acids will now get attracted closer to each other, causing more of a fold that way. And because it sort of folds that way, it has like a affinity for that new, or not affinity, but like a kind of attraction towards that other amino acid, it's going to fold a little bit differently. And so that is what a conformational shift is.
When in doubt, always think of it being like what's most thermodynamically favorable for the protein. If you have, like he said, if there's a negative phosphorus group added to a protein where there's a lot of negatively charged amino acid residues, the protein is going to shift away. It's going to conformationally change into a structure that's more thermodynamically favorable for the system, rather than keeping those negative charges close to each other because they don't want to be close to each other. Does that make sense?
Any last questions?
All right.
Yeah.
So I'm really, what is the phosphorus doing when it bonds to it? It makes the protein negative?
So yeah, like let's say you have a protein that has like a whole bunch of different amino acid residues, and it gets stuck onto something on the far right side, right? And so now,
Let's use that one.
oh yeah, I'll go over there. So if it gets added to this position right here, right? It's super negative. And there's a bunch of pluses, like positive amino acids around here. This whole structure will start to fold kind of this way, right? Because positive is attracted to negative on this side too. So this will kind of start to bend this way, and then maybe the new structure will look something like, I don't know, like this, right? Because they folded inwards, right? Or, like Aniket said, if these were all negative here, this might fold like this, because this is where the phosphate group got added, and the negatives got pushed away. Does that sort of make sense?
So this can happen post-translationally for like enzymes as well. In order for that enzyme to get into its active state, or in order for it to change its conformation around its substrate, to allow for that reaction to happen. It's just an application. But yeah.
Cool. I think that's it for our slides. All these answers for the questions are on the slides, so you can look at them on your own time too.
Yep.
Email us with any questions. My office hours are tomorrow at 1:15. Aniket's are at Thursday at 3.
3 to 4. Yep.
3 to 4, yeah. So swing by with any questions, problem set, whatever else you have. We're happy to answer them.
Yeah. Thank you guys for coming.
And if we didn't give you candy and you answer questions, come up. Or if you just want
Or
candy.
if you just want candy.